Hello, this is Dr. Bates, and we're going to be covering Chapter 11, the prokaryotes. Now, Chapter 11 goes through lots of prokaryotes. I'm going to be talking about them because students find them very interesting, and they might help you associate the organism with what it causes. But remember, on this test, 25 to 50% of the exam is the Cootie list. Okay? The Cootie list is a list of organisms and what they cause. Now, there are two types of questions that I might ask off the list. I might ask you which organism causes which disease, a matching section, and then I might ask you, give you an organism and ask you, is it a bacteria? Is it a protozoa? Is it a virus, etc.? So you need to know which category they're in, which classification, and then also what disease each organism causes. Now, don't panic, it is a long list. However, if you start looking at the organisms, some scientists were very nice and they named the organisms in ways that help you identify which organism it is. Okay, so Chapter 11 will be an overview of the prokaryotes, talking about different diseases and things that they cause. And as you listen to this lecture, you'll learn a lot of things. You'll also notice that I'm going to talk a lot about several different organisms. And to make this chapters easier, that's where the list comes in, the Cootie list, instead of knowing everything that I talk about in this presentation. So we're going to talk about the differences between, in the domain prokaryote, the gram-negative and gram-positive organisms, and then talk about the archaea. Now, in this, you must know this dichotomous key for the exam. Okay? I will ask you to give me this dichotomous key. And you need to know it because it's useful to understand how we separate the prokaryotes. So we have over here the organisms that have peptidoglycan, and then of course we separate them by their ribosomal RNA signature. And if they have peptidoglycan, then they are bacteria. If they have pseudomurin, then they are archaea. Now in the bacteria, we're going to separate them by gram-negative and gram-positive. And in the gram-negatives, we're going to see the proteobacters and the non-proteobacters. In the gram-positives, we're going to actually separate them based on low and high GC content, meaning I'm looking at the A, G, Cs, and Ts in their DNA and separating that way. Now we're going to talk about the organisms in each of these classifications as we go through. So first, the proteobacters. They are bacteria and they're gram-negative. The proteobacter comes from the name of a Greek god, which was able to assume many shapes. They're gram-negative. They are chemoheterotrophic, and they're divided into alpha, beta, gamma, delta, and epsilon groups. Now, the first one is not very exciting, except that he is 0.5% of all prokaryotes that we know of. If we took a sample someplace, you'd find him, 0.5% of the sample would be him. Or 20% of all prokaryotes in the ocean are this organism. And what's weird about him is he was just discovered using the fish technique, which I mentioned in the last lecture. It's kind of strange that it was previously unknown. It's such a huge number, but we didn't know anything about it. Just pointing out the fact that there are organisms out there that we have no clue about. Now, first in the alpha proteobacters and the human pathogens is cat scratch fever. Cat scratch fever. Usually I have at least one individual in class whose child has had cat scratch fever, and it happens, your cat scratches you. This organism is underneath its nails and it will scratch you, and after it scratches you, you will end up with a rash and you don't feel good. Almost all the diseases we'll talk about have some kind of flu-like symptoms, a malaise. You don't feel good, but cat scratch fever. Then we have brucellosis, which is a flu-like again. You get it from contaminated meat or milk, and this organism is very scary because it can survive phagocytosis, meaning your cells can eat it, your immune system can take care of it, and it still can survive there. It also can be chronic. Now, what's a big deal about it being chronic is that means that usually it means that that organism is able to hide and that we can't actually rid your system of it a guaranteed 100%. It infects cattle, goats, camels, dogs, and pigs. It's rare in the U.S., about 100 to 200 cases a year. Now, we also have, in the alpha proteobacters, obligate, intracellular, parasites. You should know what obligate means. You should know what intracellular means. And that makes these organisms, again, quite bothersome because they are required to be a part of another organism's cells in order to survive. And that means it's very hard to get rid of. So the first one is ehrlichiosis. It is flu-like. You get it from tick, flea, or mite bites. It takes 7 to 14 days for the symptoms to occur and then 3 weeks to get over it. And it's not necessarily this infection that hurts you. It's usually a secondary infection because this organism is capable of weakening your immune system. Now, rickettsias. Rickettsias are, well, they include Rocky Mountain spotted fever. This is the group of spotted fevers, and they are borne by arthropods. The spotted fever part means, if you can see in this picture, it produces little bitty blotches all over your skin. And that's because it basically screws up the permeability of your blood capillaries and you end up with them leaking, causing the spots. They actually induce phagocytosis to infect your cells, and then they reproduce in the cytoplasm. Again, that makes it harder to get rid of. So first, Rocky Mountain spotted fever at the bottom, because you know it's endemic to Arkansas, meaning it's here, we can't get rid of it. You get it by tick bite. There's approximately 2,000 cases in 2006, and about 3% of the people who get it actually die. It takes 2 to 14 days for you to develop the symptoms. Science that's saying that this may contribute to an illness later on in life. Now, there's also the Rickettsia typhi, and this one is an endemic murine typhus. It's rare in the U.S. Less than 2% untreated die. If we give you treatment, 100% recover. And then we have Prozalki, which is epidemic typhus, and it's epidemic because 10 to 60% of the individuals without treatment die. Now that's a big deal because this means if you don't know what you have a high probability of dying because we can't figure out what you're sick with, or if you were to infect a lot of people, well, we might run out of stuff. It has a high death rate, and that's scary whether we can treat it or not. Okay, so here's what ricketsias look like. So here's one of their cells, and on the outside they have a slime layer. This slime layer allows them to survive outside of a host cell until they can get back into a host cell where they actually can grow. They can only grow in a host cell. So here is the nucleus, and then all of these dark spots are actually the ricketsias. Wolbachia is an organism that lives in insects and other animals. It's used as a biocontrol. And it was really cool when they figured it out because if you look here, so we're looking at insects. Male and female insects that aren't infected have no problems. If you have a male that's infected, then you get no offspring. If you have a female infected, then you produce infected offspring. If both the male and female are infected, you produce infected offspring. And an unfertilized female can still have offspring, and they are infected. So I want you to notice, the only place that it actually produces no offspring is if only about 50% of the population has it. So as this infection spreads, it becomes less effective as a biocontrol. And then come to find out, since it lives in insects and other animals, it can actually infect people who are immunosuppressed, which makes it, well, a little scary in our current populations. All right, so here's a couple more. Now, these two are environmentals. There's nothing exciting about them, but they look different than everybody else. They have a stalk, which you can see here on this organism. Or this organism, notice this nice stalk here, and these you find, both of them you find in water, and they're just kind of cool looking, but they don't really cause any harm. They just happen to be environmental organisms. Now, Agrobacterium is also an alpha proteobacter, and we've talked about him before. He produces an agricultural disease called crown gall. Crown gall will not kill the plant unless the plant is horribly stressed. Crown gall just slows it down, and normally plants that are infected with crown gall will not produce any product. Now, Agrobacterium, however, is also important for biotechnology because since it makes crown gall by inserting a plasmid into the plant cells, inducing the tumor, it actually knows how to transform plant cells. So we can use it to change plants to get a new gene of interest. There are some organisms here that are important for environmental processes. The example here is the nitrogen cycle. We're going to talk about several as we go through this for the nitrogen cycle. At the end of the semester, we'll talk again about nitrogen and the recycling of it, how important it is. So both of these organisms fix carbon dioxide and process nitrogen. They are very important for the nitrogen cycle. Again, we'll talk about them later. Now these organisms, both of these, fix nitrogen, and they're important for plants. They actually will assist them. The one here at the bottom, rhizobium, this is a picture of rhizobium. Notice these root nodules is what they're called. These, and if you looked at those and I asked you, you would probably tell me they look like tumors, and they are. However, as opposed to agrobacterium, these tumors actually benefit the plant. So this plant that is infected is actually healthier than one that isn't, because these nodules hold rhizobium, and rhizobium will fix nitrogen in the roots. The plants get the nitrogen, and the rhizobium gets a place to live. There is a symbiosis going on. They infect legumes, and this nitrogen process is very important because if you plant legumes, you'll actually put nitrogen into the soil. Some plants like corn use all the nitrogen in the soil. So if you plant legumes after it, you will make your soil healthier. Very important. If you ever look at the label on the fertilizer that you buy for your plants, there are 3 numbers. One of those numbers is nitrogen. So it is something that we give to our plants to make them grow bigger, faster, better, etc. There are organisms in the alpha proteobacters that produce acetic acid from ethanol. The thing that Pasteur was concerned with in alcohol are these organisms. You can think of them as a negative, they damage wine, or a positive, they make vinegar. So Acidobacter or Gluconobacter, both of them take ethanol and turn it into acetic acid. Now, this is another dichotomous key. No, you don't need to know it. It's just an example of how we can break down organisms based on what we know about them. All right, so now we're in the beta proteobacters, and Thiobacillus ferredoxins, this gentleman right down here, he is used to recover waste copper and uranium. Basically, if you put him in, let's say you take the liquid from someplace that you end up with waste, copper industry, and you put him in this wastewater, he will collect the copper. And if you drain off the water, you will have this container lined with copper that Thiobacillus has recovered. And so he's very important for this. He's also very important for environmental cycles. We'll mention him again, like I said, of the others later on in the semester. Now we have 2 more. These 2 organisms, not quite exciting in a lot of ways, except what they do. So this one right here, I want you to notice, see all these guys in the chain? They actually have this sheath. Do you see it right here? This sheath. And so each bacteria cell is surrounded, and they're all grouped together and protected by this sheath. Holds them together, makes them in nice long chains, and they work together to do this. So that's what makes these guys interesting. And then, of course, this one looks familiar. You probably saw him in the microscope at the beginning of the semester. Notice his flagella on each end. And this is a spirillum. The proteobacters also involve things that make us sick. And this one right here is Neisseria. And this organism, you're probably well known for gonorrhea. So gonorrhea is also known as the clap. And if you don't know why it's called the clap, I have to tell you. And that is when a sailor would go visit women and then come back to the ship, he might come back with a clap, and it would cause pustules on his penis. And so what they would do is actually take his penis, lay it on a hard surface, and clap it with a book. Yeah, they would do that to actually break the pustules, release the pus. I'm not sure if it worked to stop him from visiting or spreading it or what, but it sounds pretty horrible. So, gonorrhea. Now, gonorrhea is commonly sexually transmitted. It's spread by contact with the mouth, vagina, penis, or anus, and it can also infect your eyes. Symptoms occur 2 to 5 days later, or a month later, or never, which is very bad about gonorrhea. Gonorrhea is very difficult to stop its spread because people who have it may never know they have it and don't take the precautions. Now, if it's urinary tract infection, we can cure it with antibiotics. If it comes in your bloodstream, it is dangerous. Now, I said a urinary tract you can cure with antibiotics. The problem is that they think about 30% of gonorrhea is now no longer susceptible to antibiotics. It's become antibiotic resistant. The UK announced in 2011 that gonorrhea was considered untreatable in the UK, and the US followed that, stating the same thing here. It no longer, if you get gonorrhea, can you just go get an antibiotic and get it treated. you may not be able to get rid of it. Now, there's also a meningitis that's caused by an organism in the same genus. It's spread by close contact. The reason it's called a meningitis is it causes inflammation to the membranes of the brain and spinal cord. 5 to 15% die. It is the #1 cause of bacterial meningitis in children, and there are several different types of meningitises, but this is the bacterial one that is the #1 cause in children and second in adults. And it tends to occur more often in the winter or spring. And I have here why. And hopefully you realize in the winter or spring, everybody's inside, the weather is what it is, and we are more in contact with each other and more likely to spread it. Also, that's when school is in session. Gonorrhea and both meningitis are capable of evading complement. We haven't talked about complement in this class, but we will later. And they also know how to steal iron from our systems. You've probably heard of whooping cough. There has been an increase in whooping cough around. And whooping cough is Bordetella pertussis, and it is an organism that is highly contagious. Once it gets into your lungs, it actually interferes with the ability of your lungs to do the silicary escalator. And this is the way that your lungs remove stuff from it. It makes the cilia, they get increased, and they basically kind of collect things or create a current, if you might think of it, and try to clean out the lungs. So what happens is pertussis will stop them from being able to clean out your lungs, the secretions in your lungs, and therefore causing the cough. Now, the cough is important because that allows pertussis to spread. It is highly contagious. There is a vaccine. But once you get it, there's no real treatment once you get the cough. Basically, we just give you medication so you don't give it to someone else, and you will have that for about 6 weeks. Burkholderia is an organism that is used for agriculture. It can cause nosocomial infections, that means hospital-acquired infections, and it can grow in disinfectants. It's kind of exciting. It infects both plants and animals, and it's very interesting for bioterrorism because there's antibiotic resistance and a high mortality rate. Zuglia, still in the beta proteobacters, this organism, well, he's used to treat sewage. That's what makes him exciting. Now, in the gamma proteobacters, we have the pseudomonads. And Pseudomonas, which we've played in the laboratory with, is an example. It is an opportunistic pathogen, both to animals and plants. It's the second most acquired nosocomial infection, or infection acquired in the hospital. It is hard to kill because it's antibiotic resistant, and it can form a biofilm, which makes it very difficult to get rid of, along with the antibiotic resistance. It causes lung damage, which means cystic fibrosis patients happens to be the ones most at risk for this causing them permanent damage, since they're already immunosuppressed and their lungs are already having issues. The whole group of Pseudomonas, so Pseudomonas aeruginosa is one example that we played in class, but there are others. They are metabolically diverse, meaning they do different things. They have been used as biocontrols. There are some that are used for bioremediation, aka cleaning up the messes we make. They can cause food spoilage, and they can produce pharmaceutical agents. They're common in the natural environment, and they have a polar flagella. And over here you can see a picture of what they normally look like. Moraxella. This organism is a commensal, meaning you can have it as part of something you already contain, a microorganism you already have, but it is an opportunistic pathogen. So when you get sick, your immune system isn't working at its top level, then you could get an infection by it. It causes conjunctivitis, notably in children, and conjunctivitis, of course, is an infection of the eye. Now we have a couple more that do nitrogen fixation in our environmental organisms. And then we have the Acinetobacters, which are environmental, except there is an organism that is capable of rapid antibiotic resistance. It is an opportunistic pathogen, nosocomial, and has a high mortality rate. Now, Legionnaires is a disease caused by Legionella pneumophila. It is an acute respiratory infection and has a hospital death rate of 50%. Hospital-acquired death rate. You can find it in streams, war water pipes, and the reason it's called Legionnaires is there was a Legionnaires meeting, and at the Legionnaires meeting they all came down sick with it. The first time we recognized it was from this group of individuals, and it was actually found in the warm water pipes in the building that they were having their meeting in. It's very interesting. It can grow in humidifiers. Humidifiers are one of the most wonderful places for microbes to grow. So if you use a humidifier, you need to keep it clean because you can actually be infusing your house with more microbes. Now we have Q fever below this. Q fever is Coxella is an obligate parasite. It infects a whole bunch of different types of organisms, and it infects, well, the organs. It is transmitted via aerosols or milk. It can take 20 days for the symptoms to come, and it can be chronic. This organism is capable of making endospores. Now here are Vibrio, and Vibrio cholera is an example of this grouping. And it is an organism that can produce toxin. It is found in poor sanitation areas during war or famine. It can infect both food and water. And it is a bioterrorism agent, and it has been used as a bioterrorism agent actually in the United States. Now here we have another organism that causes gastroenteritis, which again is going to be getting it into your system, causing a complete screw up of your digestive system. You find this in coastal waters, and you can get it from raw shellfish. Raw shellfish is always kind of scary because of the microorganisms you can actually get from. Now, this group here in the gamma proteobacters are enterics, and enterics mean that they inhabit the intestinal tracts of organisms. So you find them there. But they are of clinical importance because they cause disease. First, we have Enterobacter. It is a UTI. Normally, that's what it causes, urinary tract infection. It is a nosocomial, aka hospital-acquired, but you find it in animals, water, and soil. But it's usually more dangerous to people who are immunosuppressed. Now here's Erwinia, and Erwinia is primarily a plant pathogen, and it actually can rot tissues. Next we have E. coli. E. coli is part of your microbiota, something that you already have. But if you go visit a foreign country, it's highly likely that they have an E. coli you've never seen before, and you'll end up with traveler's diarrhea. The #1 cause of travelers' illnesses is E. coli. And it's one of the reasons why if you travel to Mexico, they say don't drink the water. You'll probably end up with diarrhea if you do because you're not used to the microbes. Now, if someone is from Mexico, of course, they're going to tell you that the water is absolutely fine, and to them it is. Again, your body gets used to what you're around all the time. Now, if a person from Mexico came to the United States and stayed for about a year and then tried to go back home and drink the water, highly likely that they would get diarrhea from the water when they went back because they haven't seen this organism for a while. Next is Klebsiella pneumonia. Of course, pneumonia tells you what it causes. It is an environmental organism. You find it in the environment, but it can also make people sick. Now here is Proteus, and we're still talking about enterics. Here we have Proteus. Proteus causes urinary tract infections. It's also known for the cause of BO, and it's really cool. Sounds great, doesn't it? Because it actually produces this swarming on the plate. See these circles? If you put the organism at the center of the plate, let's say it started out here, it will actually move across the plate, and you will see it growing and moving across the plate, swarming. So salmonella is food poisoning, of course. That's probably what you've heard of it most. It inhabits intestines, and it can make you very, very ill. Now, a particular one, Salmonella typhimurium, causes typhoid fever, if you've heard of that before. Serratia, we play with it in the lab. It can form biofilms. It is a hospital-acquired infection. However, it was at one time thought not to be dangerous because normal individuals see an infection with Serratia. Next is Shigella, and Shigella causes dysentery, and it's found only in humans. And then we have Yersinia, and Yersinia pestis is well known, causing the Black Plague. Pastorellas, still in the gamma proteobacters, are animal and human pathogens. Pastorella is a disease in domestic animals. It can cause pneumonia, fowl cholera, and sepsemia in cats. The probably most well-known organism in this genus is the Multicotia, and it is the organism that's in the saliva of the Komodo dragon that bites you for you to die or get very, very ill very quickly. Haemophilus, this genus, is named because these organisms like heme, and the Haemophilus influenzae was called this because it was thought to cause influenza. Found out it doesn't. It most likely causes meningitis and earaches in children. It can also cause arthritis, bronchitis, pneumonia, etc. If your child has an earache, it's possible that it's this organism. Another organism in this genus is sexually transmitted. Both of these organisms, or all the organisms in the genus Haemophilus, require heme and NAD, NADP to be added to the medium in order to grow. Bigoda is environmental. It actually processes sulfur, and in this image you can see it here. That's this big slimy spot. It actually can do a gliding motion and move, which is kind of interesting. Francella tularensis causes rabbit fever. You can get this by messing with a rabbit that scratches you from the wild, or a dead rabbit. You can also get it from ticks. When you end up with this, it causes a rash and a fever. It's rare in the U.S., but it is endemic to North America, so it is around. It's very interesting that it's thought to be a good bioterrorism prospect because it takes a very small amount to infect. And it's fatal in 5% of the people who are untreated, 1% treated. Now, because the fatalities are not so high, this organism for bioterrorism in and of itself is not that great. That it can infect quite easily is the important part. So what a person who wanted to make this an actual bioterrorism weapon would want to is pick the ones that are more fatal than others, and then it would be a possible weapon. In the delta proteobacters, we have different types of organisms. Most of them are environmentals. Here we have an organism that actually feeds on other bacteria. It actually reproduces in the periplasm of a gram-negative bacteria. And then we have an organism that uses sulfur instead of oxygen as an electron acceptor and is important for the sulfur cycle. So we'll mention him again later on. Now, this organism, again, an environmental. What's cool about him is that he actually forms this structure. He looks very similar to a fungus. It digests bacteria. It can have a gliding motion. And so that makes him kind of interesting that he is a prokaryote, but he is still similar to an eukaryote, a fungus. And his lifestyle is basically these spores germinate. They run around like slugs, basically. They can leave slime trails. Then they'll all congregate together, make a mound, and build this structure, making babies or spores that can germinate and go through the cycle again. So he has a very interesting cycle, and you'll see it's familiar or similar to what we see in fungi. Now, the epsilon proteobacters, there's two examples here. One that causes abortions. The other one is jejuni, which is the leading cause of foodborne intestinal diseases in the United States. The leading cause of diarrhea in the U.S. is this organism, not E. coli. Also in this group is the Heliobacter. This guy right here is very interesting because he is the cause of most ulcers. He has multiple flagella. He causes peptic ulcers, and he can be the contributing factor to stomach cancer. So if you have an infection with this guy, he's the one that causes your ulcers, not microbiology class, okay? All right, now the non-proteobacters, these guys, very simple. They're phototrophic. They can either do it with oxygen or without, and if they do it without, it's usually sulfur. So here is the cyanobacteria. I want you to notice that we have a group of them. They grow in long chains. This is what you have in your aquarium. It is not algae. It is actually cyanobacteria. Here we have a group of cyanobacteria, and each of these is a single-celled organism, and they happen to all connect together in groups of 4, and they create a glycocalyx all around the whole grouping. Now, the purple sulfur bacteria, they're going to use sulfur, and they're just cool because they do this. They're very important for the sulfur cycle, and they're multiple colors due to their use of sulfur. Now, the firmicutes are gram-positive bacteria. They have low GC, and there's a couple things that are interesting about this group. The first one in this group to talk about is Clostridium. Clostridium is an endospore-producing organism, and you, of course, probably know of it as botulism. So Clostridiums are resistant to heat and chemicals, and they look like tennis rackets. They are obligate anaerobes, and botulism being a very important one. Of course, tetanus. Many of you have had your tetanus shot. It is also a Clostridium. And C. diff, if you've heard about that, it is a microorganism that we are finding very important in hospital-acquired infections. If you use antibiotic on people, it will hurt other organisms, but not this one, and it will then be able to overgrow. So while treating one infection, you actually cause this one to, well, take its place. There's also Provenges, which causes gas gangrene, a common foodborne diarrhea. Now, this guy right here is also in the same category, but I want you to notice what he looks like. What's so cool about him is he's huge. You can actually see him with your just naked eyes. He has 25 times more DNA than humans. He's still a prokaryote. He has no membrane-bound organelles. And we said before that the reason the cell size is because you have to move things around. So this organism has proteins that actually moves nutrients around in it. And it is a microbiota fish. Now, who decided to look at what bacteria are growing in fish? I don't know, but they opened up a fish, found this organism, and it was very interesting because he's quite large for a prokaryote. So the genus Bacillus are endospore-producing rods. We have Bacillus cereus, which is a probiotic, can also cause food poisoning. We have Bacillus anthracnus, which causes anthracnose. You've heard of it before. Of course, it can infect cattle, sheep, horses, and humans. It's used for bioterrorism because you can put the spores in an envelope, mail it to somebody, you can inhale the spores, and you get flu-like symptoms. Anthrax is actually found in nature. It is not something that's brand new. It's not something that was created just for bioterrorism. If you've ever seen a deer on the side of the road and its whole body is like puffed up, it's very possible that it's infected with anthrax. We're talking about a dead deer on the side of the road. Now, Bacillus thuringiensis is the organism that was used to make BT corn. So this is for a process of taking a gene from one organism, sticking it into a plant, and allowing that plant to be able to, well, destroy insects that try to eat it. Staphylococcus, we've heard about this group before. Staphylococcus, they have grape-like clusters. Staph aureus is named for its color. It's actually golden color. However, when we look at it in the microscope, we don't see this. It can handle high osmotic pressure and low water, which means it can survive on your skin. It actually produces toxins, and it causes toxic shock syndrome. Many of the ladies in class will actually have heard of this organism before because it is on the tampon labels. It is possible that using tampons can cause an overgrowth of Staph aureus and cause toxic shock syndrome. Staph aureus produces toxins that overload your immune system and make your immune system just like override itself or do more than it should, overreact. And so you end up with your own immune system, well, hurting you, possibly causing death. It can also cause food poisoning, and it is antibiotic resistant. We'll talk more about Staph aureus later. And then, of course, Staph epi, which we've played with in class. It is a skin microbiota. It is good for you to have, but it can be a pathogen of the immunosuppressed. It can cause sickness if your immune system doesn't keep it in check. Now, lactobacillus, these organisms are aerotolerant anaerobes. They lack the electron transport chain. Lactobacillus is important for sauerkraut, pickles, buttermilk, yogurt, different types of food production. It is a microbiota. It is important for you to have. But also streptococcus, well, that organism is pathogenic. It is actually capable of destroying phagocytotic cells. It can kill macrophages. And it will digest connective tissue in an infection. So it's actually known as a flesh-eating microorganism. Some non-pathogenic species are used for dairy production. Now we have Enterococcus. This organism is a nosocomial urinary tract infection. You can get it in surgical wounds. It is antibiotic resistant. And then we have Listeria, which you've probably heard about before. It's really concerned in dairy refrigerated substances that have not been properly maintained. You can get Listeria, and Listeria is well known for causing miscarriages. Now, Mycoplasmalis is a very interesting organism because this genus is wall-less. They are also pleomorphic, meaning they have more than one shape, which is two things that actually break the prokaryotic rules. However, everything else about the organism makes it a prokaryote. There is one pneumonia that will actually cause pneumonia. And down here you can see the single organisms here and the shape of them. And then here shows this same organism. You can see single organisms here, and then this huge glob will produce more fragments of more organisms. Now, the Acinetobacters are gram-positive bacteria. They are high GC. And we have actinomycetes in the ubiquitous lab where we collected samples. If you had this fuzzy thing, but it wasn't fuzzy, it just looked like it made swirls on the plate, and I told you it looks like a fungus, but it's not. It was an actinomycete. You find him in the soil. He's also in the mouth and throat of humans, and here's what he looks like over here. Now, Corneobacterium is an environmental. Diphtheria is probably an organism you've heard of before. Diphtheria causes upper respiratory tract infections. Franca is another environmental that is nitrogen fixing. Gardnerella has a species that is an STD. And then Mycobacterium, of course, tuberculosis and leprosy caused by Mycobacteriums. They are acid-fast, drug-resistant. They are pathogens. If they're slow-growing, then they are dangerous. If they're rapid-growing, then they just happen to be environmental and not of concern. Now, Nocardia is another soil organism, but there is a species that causes infection. Propionibacterium in this genus is the organism that causes the fermentation of Swiss cheese, but it also can cause acne. Streptomyces looks like curly fries connected together. That's what it looks like over here. It is the most common soil microbe, and it's the reason that soil has that musty odor. It produces about 2/3 of our known antibiotics. A very important microbe in the soil. Now, the non-proteobacters, they are special. They usually have something strange about them. The first one here actually has an internal membrane around its DNA. Another broken rule in prokaryotes. But he has nothing else. Everything else makes him a prokaryote. His ribosomal RNA, he's found in fresh water, but it does make it interesting that he has this kind of pre-nucleus. Chlamydias. Chlamydias at one time were thought to be viruses because they couldn't be grown in a petri dish. There are only 3 species. Each of them causes disease. We have the first one, which is an STD. It causes pink eye. Pink eye, if someone says you have pink eye, it's usually chlamydia. It isn't always. There are other organisms that cause infections in your eye. But pink eye is normally caused by chlamydia. And you've probably heard that if somebody has pink eye, don't look at them in the eye because you'll catch pink eye. And every time I say that in class, everyone laughs because it sounds funny. But you know what? The answer is yes. If you look at someone who has pink eye, it's possible you'll contract it because what happens is pink eye causes their eyes to water. So when they blink their eyes, it's the same as sneezing. They're actually going to project that liquid out of their eyes, and if you're looking at their eye, if you're close enough to look in their eye, then it's highly possible that that liquid can be splashed from their eye into yours, and you can get an infection. An infection that you find in bird droppings, and it causes parrot fever. The chlamydias, you can't grow them in a petri dish because they must be inside of a cell. So what they do is they produce an elementary body. That's basically a waiting stage until it infects a cell where it will turn into a reticulate body, duplicate, multiply, turn back into the elementary bodies, get released from the cell, and then go and infect another cell. Spirochetes are an interesting group because these are the ones that have the axil filaments, so they actually can curlicue into tissues. So we have Lyme disease that's transmitted by ticks and lice. We have a disease, leptospira, that causes redness of the hands, and it can come from contaminated water. And then we have syphilis. Syphilis is very scary because it can pass through the placenta. So if you're infected with syphilis, you can pass it to your offspring because it can actually burrow through tissues. Bacteroides, these guys are anaerobic. They are found in the mouth and large intestines. They are a microbiota, and they're not bad, except they can acquire antibiotic resistance. Now, it doesn't matter because they don't make you sick, but what does it mean if you have one organism in your body that knows how to overcome antibiotics? Now there's another one. This guy can actually degrade cellulose in soil. Becomes very important when we talk about biofuel because they want to take this organism who can break down cellulose and then use another organism that can turn that cellulose into ethanol. Remember, broken down cellulose is going to be single sugars. The catch is that it's not quite as good as we would like it to be. So there is current biotechnology going on to figure out how this organism degrades cellulose and use that for a product. Now, Fusobacterium are found in the mouth, and they may be involved in dental diseases. They're not exactly sure. And something that's very important about these organisms that you find in your mouth is they form plaques. Plaques, of course, are similar to biofilms. And there is current scientific knowledge that says plaques in your mouth may also be related to the plaques you find in your blood vessels. Now, the domain archaea. This group, very simple, short, and to the point because they are extremophiles. We have the hyperthermophiles, the methanogens, and the extreme halophiles. They like it hot. They don't like oxygen. That's the methanogens. They produce methane gas. They produce swamp gas. Or they can handle high salt conditions, the haliophiles. Now, the methanogens are very interesting because they actually are used to see if life on Mars could exist because Mars doesn't have very much oxygen but has lots of methane. If methanogens can live in a fake Mars environment, then it is very possible there could be organisms living on Mars. All right, so the last thing is just to talk about microbial diversity. We talked about them, and you'll notice in each group, they're good guys, bad guys, environmentals, things that kill people. Well, why don't we know about all the organisms that are out there? And the answer is, well, mainly it boils down to because microbes are so different in size and really so small. that it's difficult to know where they're at. For example, in 1 gram of soil, basically a spoon of soil, PCR says that there's 10,000 bacterium. However, we haven't been able to culture them all. And if we can't culture them, we don't know anything about them. We can't identify them. So microbes aren't identified because they haven't been cultured. They need something in particular to live. They have some kind of complex food chain that requires them to get food from another organism. There is a giant food web in the soil, in the environment. And until we can, well, grow them, then we can't figure out how they work or why they're important. And the ecological role becomes a very important issue in these places where maybe there's been an oil spill we're trying to clean up. Maybe if we knew what microbes are supposed to be there, we can help the environment get back into its native state. So we talked about all these different organisms. Now, I'm not going to ask you about all these different organisms on the test. You do need to know, like Proteobacter, there's alpha, beta, gamma, delta, and epsilon. In the non-Proteobacters, you find the photosynthetic organisms. That's what I want you to know, just the basics. Don't know what each and every organism does, because that's your cootie list. All right, well, that's the end of it. If you have any questions, please contact me and let me know.